TWiM listeners, do you have science you want to share? Microbeworld.org is the best place to post your microbiology-related news articles, pictures, videos, papers, and more. Sign up for a free account at microbeworld.org/join and start reaching thousands of microbiology enthusiasts just like yourself.
This Week in Microbiology is brought to you by the American Society for Microbiology at microbeworld.org/TWiM. This Week in Microbiology, episode number 81, recorded on June 19th, 2014. Hey everybody, I'm Vincent Racaniello, and you're listening to TWiM, the podcast that explores unseen life on Earth. Joining me today from Ann Arbor, Michigan, Michele Swanson.
Hello everybody.
How are you, Michele?
Doing good, doing well.
You have great weather out there?
Yeah, we had some terrific thunderstorms come through in the last 2 days, so now it's pretty cool, mid-70s.
Well, here it's all cloudy today. It's going to rain. It's like 23 Celsius. Not a great day.
Huh.
Also joining us today from Charleston, South Carolina, Michael Schmidt.
Hello, everyone.
Did I get the city right?
You got the city right.
I'm not used to saying Charleston for some reason. How's the weather there? Chamber of Commerce?
No, it feels like we're in the Gulf of Mexico, or it feels like Houston. It's hot and sticky.
We're in the summer.
Yeah.
Actually, officially, we're 2 days. It'll be summer, right? June 21st.
Yeah, it's about 35 degrees Celsius.
Wow, that's hot.
And the humidity is condensing on my windows outside. So
Yikes.
And my office is not that cold, so.
Wow. Well, it is summer, and before you know it, it'll be over, and we'll be shoveling out of the snow.
That's right.
You know, the seasons just go zipping on by. All right, today we have two papers for you that both have to deal with iron. My colleague Dixon Despommier told me yesterday the amount of iron in a human is equal to one nail. Although he didn't say what size nail.
A penny nail?
Yeah, it's exactly. It could be tiny or it could be large. But that's kind of a nice visual thing, right?
Yeah.
One nail. And this one paper that I'm going to do said the concentration of iron in body fluids of humans is 10 to the minus 18th molar. That is pretty small.
Tiny.
But bacteria love iron.
Oh, yes.
And both of our papers have to do with iron today. Coincidentally, you may think that we planned it this way, but I just was scanning titles, and these two stuck out, and it turns out they both have to do with iron. Michael, do you mind if I take the MBio paper first?
Go ahead.
It's a bit shorter than yours. And
yeah, it's a bit shorter than mine.
Well,
we may get stuck in the weeds.
You know, if you go on too long, Michele and I will just leave and let you record.
Take a nap.
Take a nap. Thank you.
My paper.
I feel so loved.
No. We're kidding. We never sleep when you talk. My paper was not mine, but the paper I will discuss was published in MBio on 10th June 2014. It's called Bacteria Present in Carotid Arterial Plaques are Found as Biofilm Deposits, which may contribute to enhanced risk of plaque rupture. And the title, it says everything. That's all you need to know. The authors are Bernard Lanter, Karen Sauer, and David Davies. They're at Binghamton University in Binghamton, New York. Has either of you heard of Binghamton University?
Oh,
yes.
You have? It's not the same as SUNY Binghamton, is it?
It used to be SUNY Binghamton, and then the SUNY systems all renamed their daughter products. Rather than SUNY Binghamton, it just became Binghamton.
Oh, okay. So I knew SUNY Binghamton very well.
Just like SUNY Stony Brook became Stony Brook University.
Why don't I know this? I guess I'm living in a cave.
Now you do.
I'm living in a cave.
Or I got it wrong.
No, well, if you got it wrong, we'll get letters.
That's right. And Elio will be most impressed that I got it wrong.
I don't think so. All right, this paper is all about atherosclerosis, which is a disease of arteries, as you could guess from the title. Probably everyone's heard of it, that happens when you get accumulation of fat and calcium within the arterial wall, and the arteries lose their elasticity. Lots of complications associated with that, including high blood pressure, as you might imagine. But one of the main problems is that these deposits form what are called plaques or atheromas in the artery walls, and they can rupture. And it turns out that 76% of all fatal coronary thromboses, those are clots that go into your heart and cause problems, arise from rupture of these arterial plaques.
And could I just interject?
Yeah, you could.
Atheroma is Greek from the word atheria, meaning gruel.
Who has an old-fashioned phone?
That would be me.
Really?
Yeah. Are we going to delete that?
No, I love it. I want to hear you make the phone call. Is it going to stop?
Eventually it will stop.
Okay, we'll wait.
So atheria is Greek for gruel.
Gruel?
So atheroma is a lump of gruel.
It's very great.
So that's what's forming on the inside of your arteries.
So some of the first people who looked at them must have said, oh, this looks like gruel that I had for breakfast. Yeah, that's great. Greek atheroma. Now, it's previously been shown that these plaques, these arterial plaques, seem to have bacteria in them. And I found a paper while looking around from 2005 from the European Journal of Clinical Investigation, where they identified various bacterial DNAs in coronary arteries, and they actually found a whole bunch of them, and they seem to be a subset of what's in your mouth, oral bacteria, because as you'll see, this paper talks about a certain kind of bacteria being present in these plaques, and I was wondering how they got there. So maybe when you brush your teeth, we've talked about this before, you introduce bacteria into your bloodstream, and those of you who are healthy, that gets cleared right away, but maybe that's where these bacteria come from that eventually lodge in plaques.
And if you brush your teeth twice a day, you're bacteremic. That is, the bugs in your mouth wander through your bloodstream for about 8 hours.
8 hours?
Wow. 4 hours in the morning and 4 hours in the evening until your immune system clears them out. And it's principally your liver and spleen and your macrophages that go up and hoover them all up.
Which is why some people, when they're at the dentist, are given antibiotics before dental work.
Really?
If they're at risk, if they have a heart murmur, they'll give antibiotics prophylactically.
Interesting. Well, the idea in this paper is you will see there are bacteria in these plaques. So maybe if you're developing a plaque and you brush your teeth, some of the bacteria may lodge in the plaque. The plaque may act as a filter of some kind, and the bacteria get there. So it's best to not have plaques to begin with, I guess. And I don't know what controls that: diet, genetics. Anyway, okay, so we already know that there are bacteria in arterial plaques, and they say, well, maybe they're biofilms. They don't have any evidence for that yet. Maybe they're biofilms, in which case maybe when the bacteria disperse, or they call it a dispersal event, which I kind of like. So the biofilms sometimes disperse. As Michael talked about a while ago, the biofilms of strep in the lung, right, get dispersed by influenza virus infection.
Right.
And so here they're thinking the biofilm in a plaque gets dispersed, maybe by some nutrient that becomes available. The bacteria go up, there's food around, they let go of their hands, and they go into the bloodstream. Of course, to let go of their hands, that's a metaphor. The bacteria have to secrete enzymes that digest the matrix that's holding them in this biofilm. So that could cause damage, and maybe it makes the plaque rupture. So collateral damage, this is their idea for what's going on. Now, why would all of a sudden nutrients be available? Well, they're thinking of iron, which is limiting. And they're thinking, well, stress is a known trigger for acute cardiovascular events. So work-related stress, what else is there? any kind of stress. You get stuck in traffic, you get stress. It's funny, I was with some physicians Saturday night, and one of them said, Vinny, if you keep commuting, you're at high risk for cardiovascular disease, because every time you get stressed, you make hormones, and they raise your blood pressure, and eventually, as you get older, your heart's not going to like that anymore. And then Monday I come in and I read this paper, and the idea here is that, again, stress, one of the things that happens in stress is that you get increase in plasma norepinephrine or noradrenaline, right? And one of the things the norepinephrine does, it binds to ferritin, which is an iron carrier protein, and causes release of the iron. And then any microbes that are around, we have iron. Let's release from our biofilm and proliferate as planktonic microbes. So stress, norepinephrine, more iron, bacteria get released from the plaque biofilm, and you get a cardiovascular event. It's a great series of events.
And they need the iron to make more DNA, because you can't make more bacteria because the iron is essential cofactor in the DNA polymerases, along with magnesium that every card-carrying molecular biologist knows about.
Is there anything else that they need iron for, Michael?
The electron transport chain. We've all heard about the evil iron-sulfur proteins when we were memorizing the Krebs cycle as novices in biology. So iron-sulfur proteins, cytochromes, all of those other things, need iron, and it's there are trace elements are typically in the if you will, picomolar concentrations, and in the cell, the iron is typically found in micromolar concentrations, whereas most of the chemicals used for chemistry by bacteria are in millimolar concentrations. So iron's in this sort of in-between category.
So, Michael, if you, in your gut, are there iron-binding proteins that keep the iron low so that...
That's the next paper. We're going to learn about that in the next paper.
All right. So
unless you want me to go into...
Not yet. It's
not
fine.
Yet.
I just can't help asking questions. All right. So basically what we're saying here is atherosclerosis could be a biofilm-associated chronic disease. All right. So what they did to test this idea, they got samples of diseased carotid arteries. Those are the ones that are on either side of your neck, right, that go from your heart up into your brain. Is that right?
That's right. Yep.
And from 15 patients with advanced atherosclerosis, so these carotid arteries have plaques in them, and they did PCR, polymerase chain reaction, looking for 16S ribosomal RNA, which would be signatures, of course, of various bacteria. They found bacterial ribosomal RNA in all 15 samples. 5 of them had 10 to 18 unique signatures, unique kinds of bacteria, and 8 of these were common to 5 of the patients. So 5 of the patients had 8 of the same ribosomal RNA signatures, and some of them had signatures that were not present in the others. But all of them had some kind of 16S ribosomal RNA associated with these diseased carotid arteries.
And they don't have to be diseased. There was one of the early trials that looked at these atheromas. Mm-hmm. They looked at motorcycle victims in Sweden, if I'm remembering the paper right. And the motorcycle riders that actually died from a horrific motorcycle accident were in their teen years and early 20s. And they were able to isolate bacteria out of these plaques that were beginning to form in the carotid arteries of young people. So you just don't have to be ill. The microbes are constantly trying to attach themselves to us.
But you have some kind of a plaque that gives them a foothold, right?
Yeah.
I think that's the paper from Finland that I have here from 2005. Yeah.
It probably is.
Because they say they looked at the coronary arteries 3 days after death. Yeah, within 3 days after death, yeah. Okay, so in this case, though, in this paper, they looked at diseased arteries. They found ribosomal RNA signatures in all of them. They previously had been, in another paper, they had been able to culture Pseudomonas aeruginosa from a carotid sample. In fact, 6 out of the 15 of the samples in this paper had Pseudomonas species. 5 of them had Pseudomonas aeruginosa. Okay, they don't talk about what the others were, but this is the main one that they're focusing on. So then they did fluorescent in situ hybridization, which has the nice acronym FISH, to see what these bacteria look like in the carotid artery. So they do sections, and then they stain them for nucleic acid, and they can see what they look like. And they see what they call extensive microcolonies in the area of tissue damage of the plaque in all of the 5 samples that they examined. Now, I have to admit, I don't have a lot of experience looking at biofilms in blood vessels, so I have to take their word that these are biofilms. But it looks reasonable. So that's the evidence that there's a biofilm, some pretty good microscopy. The last experiment, they asked, can these biofilms be dispersed by iron? And of course, they can't do that experiment in these samples because they're non-living anymore, but they have they set up a biofilm in a silicon tube, so thin silicon tubing in the lumen of the tube. They put medium and they allow the bacteria to attach to the wall, and then they can measure they can take the fluid that comes out and measure the number of bacteria in it. And when they add iron, they see that bacteria are released from the wall, or released from the biofilm. And more iron you add, the more release you get. It happens in about 20 minutes. So at least in culture of Pseudomonas aeruginosa, you can get release of single bacteria from the biofilms. They also do an experiment where they add transferrin that's been preloaded with iron. So again, transferrin is an iron carrier protein. They add that plus norepinephrine, which will bind the transferrin and release the iron. Indeed, that causes release of bacteria from these artificial biofilms. And if you just add the iron transferrin alone or the norepinephrine alone, you don't get any release. So their idea is on the right track, I think, that these biofilms could be released by increases of iron concentrations. So they make a nice summary in the discussion. They say there are 4 characteristics of biofilm bacteria. One is that they adhere to either a substratum or a surface. 2, that they aggregate in clusters encased in a matrix. 3, they're confined to a particular site in the host. And 4, they're difficult to control with antibiotics in the biofilm, even if they are sensitive in planktonic stages to the antibiotic. So they say this study has shown, at least in this case, 1, 2, and 3.
And the transferrin experiment, for those who may not know how tightly transferrin at a physiological pH of about 7.4 has a binding affinity of 10 to the minus 23 molar.
Wow.
So that's an incredibly high affinity binding site for iron 3. But, you can tinker with the pH, but it really doesn't matter in a biological system, because if you lower the pH too low, you're dead. So 10 to the minus 23 at pH 7.4 really gives you an idea of how important this iron is to their paper.
It's interesting that norepinephrine can cause that to be released. Must do something very interesting to the transferrin, right?
Yeah.
And the release would have a number of implications. As they speculate, when the bacteria switch into that swimming mode, they likely release a number of enzymes like elastase and proteases and phospholipases, which are known to degrade host molecules and tissue. So they think that could contribute to expanding the damage at that local site. Right. And then once the bacteria are free into the bloodstream, you could imagine they traveling through arteries, and then if they come across another site of damage, they could latch on and start a new biofilm. So you could have more extensive disease then as a result. As a result of commuting in heavy traffic, Vincent.
I was just going to say, it's quite clear I need to stop commuting. Absolutely.
You just need to get a driver.
Yeah, that would be cool.
So I had an opportunity to talk with David Davies, the PI on this paper, and also Bernard Lanter, who was the PhD student who did the work, and they told me a bit more about their research team. It was a collaboration with another investigator there in their department, Karen Sauer. So both Karen and David have expertise in biofilms. They actually were both postdocs at the group out in Montana, the Biofilms Research Center in Bozeman, Montana. And they were able to approach the United Health Services, which is the big network of hospitals in that region of New York State. And because UHS has a mandate to contribute to intern training, they're interested in contributing to the research mission at those health centers. And so Davies was able to talk with some cardiovascular surgeons who normally treat people that have occluded arteries. And so to treat them, they actually resect. They go in and cut out sections of these damaged arteries. And typically they'll use half of it for pathology, and the other half they would save, and if they didn't need it, they'd throw it out. So the microbiologists were able to get those patient samples.
Nice.
And then Bernard Cantor, who actually was an undergrad at Binghamton, did some research in his senior year in David Davies' lab, got interested in microbiology research, and then he's the one that did the fluorescence in situ hybridization analysis on these samples. And I had a chat with Bernard, and he described that this microscopy was all new to him. He actually was a chemistry major as an undergrad. And at first, when he saw these images with the little clusters of bright staining, he wasn't sure what he was looking at. But after consulting his anatomy textbook and looking at what arteries look like in the three different sections, he got quite excited and realized that they were probably onto something. So they hope to test some of these models maybe in a mouse model, because there are a lot of experiments that, as you point out, are just not possible to do in the patients.
Yeah, I was thinking about that. It would be nice to see this in mice next, right? I guess you could somehow induce plaque formation in mice.
Yeah, evidently that there's a mouse model. They're deficient in ApoE, which is important for cholesterol metabolism, and so you can generate plaques in those mice, and then that would allow them to test some more specific parts of their model, such as during dispersion, do the Pseudomonas in fact secrete some of these enzymes that cause tissue damage locally, etc.
So in the long term, if this turns out to be true in humans, that humans have bacteria in atherosclerotic plaques, and that's part of the rupture, is that something we could treat?
Absolutely. I'm not sure how.
Just don't brush your teeth.
No, you don't want to go there. You want to effectively find the magic bullet that will keep the bacteria, before they get to a critical level, to decamp more often.
Yeah.
So you don't have, with atherosclerotic disease, I guess it's all about disease burden. And the more plaques you have, the more you're at risk. So I guess if you could prevent the plaques from forming, which is why everyone is telling us to eat the right kinds of oils, principally fish oils and these other things that retard or lower the likelihood of forming these atherosclerotic plaques. But it's all about wounding your vessel and C-reactive proteins involved, and I get the lecture every time I go to the doctor, and they look at my blood levels and they tell me, oh, this is what you need to do.
It's not easy in the South. But I was wondering if they say that moderate exercise increases the amount of norepinephrine. So I wonder if moderate exercise causes that release, causes an increase in iron, and you're getting shedding of the bacteria from these biofilms, and then they would be cleared if you're otherwise healthy. So maybe moderate exercise prevents those biofilms from getting too large.
I think that's as good a hypothesis for exercise that I've ever heard.
Moderate
from a molecular perspective.
That means golf, moderate exercise, right?
I do walk too. I don't carry my clubs, but I pull them in a cart.
That's okay. I was going to say you don't have someone carry them for you, right?
No.
Good. Pulling the cart is good. These are all interesting studies to be done. Do you know, Michael, if you can do some kind of imaging and see plaques on a human? Or is that too small?
I think you can. I'm trying to remember some of the seminars I've attended with my friends in vascular with the confocals and whatnot. I think you can if they light things up, but I'll have to go do some literature hunting.
Okay. Anyway, a nice paper. Thanks for talking with them, Michele. That's pretty neat.
It's a pleasure.
And I think this is cool stuff that's going to lead to something down the road. So,
and the parallel might be with Helicobacter pylori. We first learned that bacteria are actually contributing to ulcers. So now, if it's true that bacteria contribute to the severity of heart disease, that's good for our field and promising as a new avenue of therapy.
It's so interesting that they're great for us and they can be bad for us.
Well, Pfizer, if I'm remembering right, did a clinical trial in which they were using to try to clear chlamydia. And there was this was when chlamydia was named chlamydia pneumonia. Now it's been renamed chlamydiophilia pneumoniae. That organism was strongly correlated with the plaques. And remember, chlamydia goes through that elementary body, and it goes through this very complex life cycle. And it's a obligate intracellular parasite, and it grows in the epithelial layers, specifically in arterial vessels. And they tried to address the clinical question, and they never really came to a good resolution that chlamydia was cause and effect in cardiovascular disease. And so it may be a combination of this, that now we can overlay this epinephrine component to it, and they may wish to go back and review their data and look at, because they were going to treat folks with antibiotics to see if they could reduce the disease burden or reduce the risk of having a fatal infarction as a consequence of the chlamydia being in the ascending coronary arteries.
But if they're in biofilms, if the bacteria in biofilms, they're really hard to treat with antibiotics.
Yep. Right.
You'd actually want to treat with iron first to get them come out of the biofilm and then hit them with the antibiotic.
Yep, which is why I think this ended up in MBio. I think it's something new and
provocative.
Absolutely.
Yep. All right, let's move on to your paper, Michael.
Well, this one is a real textbook case of a great paper in the journal Nature, and its title is "Inverse Agonists of Estrogen-Related Receptor Control Salmonella typhimurium Infection by Modulating Host Iron Homeostasis." And it's by a group of authors out of the country of Korea. And the first author on the paper is Dan-Koo Kim, and he is at the National Creative Research Initiative Centers for Nuclear Receptor Signals in Hormone Research Center in the School of Biological Science and Technology at Chonam National University in Gwangju in the Republic of Korea. He has a large number of co-authors, and the senior author is Wang Sik Chu, and he's also at the same institution as the first author. First up, this is only got 4 figures in the paper, but really there's about 40 figures in this paper. But they're really easy to decipher. And as with all Nature papers, you have to read each figure, in pieces. You read figure 1A first, and you digest the significance of it, and then you move on through the very complicated figures. And we'll get to that as we get to the data. But I'm going to give you the bottom line up front first, or the bluff, as they say. Estrogen-related receptor, gamma, which they abbreviated EER and then with the symbol gamma, is a major transcriptional regulator of Salmonella typhimurium-mediated induction of hepcidin. Now, I didn't know what hepcidin was, and rightly so, because it's a peptide hormone that's produced by our livers. And it was only discovered in the year 2000, and it appears to be the principal regulator of iron homeostasis in humans and other mammals. And what the hepcidin does, and this is key to understanding what this paper is all about, is hepcidin functions to regulate, which in this particular case means inhibit, iron transport across the gut mucosa. So what this does for us is when we eat a bunch of iron in our diet from green leafy vegetables, it prevents iron overload in our body, which would effectively let the bacteria multiply and kill us dead. So evolutionarily, hepcidin is really important to control just that we get the right amount of iron. And then most importantly for Salmonella typhimurium infections is hepcidin inhibits the transport of iron out of macrophages. And this is where we hide our iron in our body. We lock it up inside our macrophages so that it doesn't wander aimlessly to feed those evil biofilms. And so hepcidin inhibits the transport of iron out of our macrophages. And many of you know that when we ingest Salmonella, it leaves our gut and our macrophages scarf it up. And then because of the wonders of type 3 secretion and all the wonderful things that Salmonella is able to do, it actually can grow inside the macrophages. Salmonella is what I was always taught is said to be one of these facultative intracellular parasites. It can grow as a free liver, or it can grow inside both non-professional and professional phagocytic cells. And a macrophage happens to be a professional phagocytic cell. So back to the bottom line, after we now know the importance of the induction of hepcidin, and its expression leads to the alteration of host iron metabolism. And then what they describe in this paper is they discovered an inverse agonist of this EER gamma receptor, and it's got a name, GSK 5182. And I'm just going to call this the antibiotic because that's what this inverse agonist does, is it has an antibiotic effect.
But it works on the host.
It works on the host. I was just getting to that. And the consequence is the antibiotic is on the host, so it's not against the bacterium. So if you're beginning to think about new strategies or new targets or chemicals of antibiotics, traditionally we always think of antibiotics acting on the bacterium, but this antibiotic actually acts on the host. And it effectively is a new magic bullet because it plays the game with iron. So the way they do their experiments is pretty much the way we get a Salmonella infection. We ingest Salmonella. And so what they typically did is via gavage, where they actually force-fed the mice either 10 to the 8th or 10 to the 9 colony-forming units of Salmonella typhimurium per animal. And this is uniformly lethal to animals if they get at least to the mice, the Salmonella. And so the results that they're generating are specifically for Salmonella, but you can think about this also being generalizable to others of these facultative intracellular parasites, such as Yersinia is a good one, the plague. Chlamydia is another one that they introduce in their narrative. But then, of course, there's other things like Legionella. And it all facilitates around this whole issue of iron and how iron is controlled by the host, and the host is very stingy with this. And so the way they begin to introduce their experiments to us is they examine the serum iron levels and the expression of this estrogen-related receptor, gamma EER, in the livers of mice. And the mouse that they used was the classic C57 black. And they had a virulent strain of Salmonella, SL1344. which is one that is classified that can infect the intestinal epithelial macrophage, and then that macrophage escapes the intestine through the mediastinal lymph nodes. And I put in the show notes for Ray a wiki diagram of where the mediastinal lymph nodes are, because it's always good for microbiologists to remember the infections are in people and the plumbing does matter. And that's actually one of the take-home messages associated with one of their figures is the mediastinal lymph nodes, and they did a cute experiment with that, but we'll get to that in a minute. So we're going to take you through the figures, and the first figure up is they did quantitative PCR where they measured the receptors, and so they're measuring receptor production, and they looked at EER gamma, and then they looked at another estrogen related receptor, alpha, and hepcidin, and then they fed the animals 10 to the 8th or 10 to the 9 Salmonella typhimurium, and they compared it to the control, and they asked, do these receptor levels go up from the liver from the livers of those animals? And the answer was hepcidin levels went up and EER gamma levels went up. So that's panel A of their figures. First thing, quantitative PCR. The Salmonella causes this receptor to rise and hepcidin to rise, but EER alpha did not. So these findings suggested then to them that there's an inverse correlation between serum iron levels, ultimately because of what hepcidin does, and the numbers of Salmonella typhimurium. Panel B is actually where they do traditional viable counts, where they're isolating the bacteria from the liver, the spleen, and the mediastinal lymph nodes, and they're actually showing post days of infection. And they show as the numbers of microbes go up, and they're measuring it as colony forming units per gram of tissue, you can see that it's significantly higher in the liver and the spleen and the mediastinal lymph nodes. So showing you that the Salmonella are moving out of the gut into circulation, and the serum iron levels are dropping like a stone. So that's panel A and panel B of that first figure. So then they're looking at this whole issue of inflammation. And they're specifically looking at IL-6 because they had some knockout animals to IL-6. There's a long, involved explanation going through much of their supplemental data that I probably don't have time to take us through. But needless to say, IL-6 is secreted by T cells and macrophages to stimulate the immune response during infection and after trauma. And IL-6 plays, as we all know, an important role in fighting infection. And it's very important for clearing strep pneumo infections in mice. So they do the oral administration of Salmonella typhimurium into the knockout animals. So these are knockout animals with that don't make IL-6, and they do the same experiment. They look at serum iron levels, and they ask the question in the control group versus the wild type. Remember from the first set of figures I described, iron levels go down when you give Salmonella. And what we see in the knockout animals is they don't, and they're missing IL-6. So, or if you will, the induction of EE, the estrogen receptor gamma. So then, as they learn from these knockout animals, the Salmonella typhimurium levels were lower than those infected in the wild-type mice at 3 days. And they found similar numbers of Salmonella typhimurium in the mediastinal lymph node. Again, this particular panel, which is panel E, but it's specifically teaching us something about the plumbing and the fact that IL-6 does not control bacterial entry from the intestinal lining into the residual macrophages, because the IL-6 knockout the bacterial levels in the mediastinal lymph nodes were equivalent between the wild type and the knockout, while in the other areas that they harvested, looking at the viable counts, specifically the liver and the spleen, the knockouts were effectively looking like the control in the mediastinal lymph node. So again, it's showing you that this take-home message from these first, if you will, 5 figures, panels A through E, suggests that Salmonella typhimurium-mediated IL-6 induction affects this magical expression of hepatic EER gamma and the hepcidin levels, thereby altering serum iron levels, and thus leads to this proliferation of Salmonella typhimurium in the spleen and livers of mice. So then, in the next set of figures, they elected to investigate the molecular mechanisms underlying the regulation of EER gamma expression by this ILS signaling pathway, where they ask, what effect does IL-6 have on gamma expression by both time and dose? And the take-home message from this complicated figure 2, and there's effectively 1, 2, 3, 4, 5, 6, 7, 8, 8 figures here, A through H, is the take-home message is EER gamma induces hepcidin messenger RNA expression and regulates iron metabolism. And if Elio were on the line, he would kill me if I went through each of these figures to the same detail that I went through the first one. So I'm going to spare the listeners that.
You learned from him well, huh?
I learned from him well. But I really don't want to shortchange the authors, because this is an incredible amount of elegant work. And each one of these figurettes is teaching us so much about the biology of Salmonella typhimurium infection as it moves from our guts to our livers and spleens. And it's teaching us how our immune system, through this IL-6 signaling, is actually doing its magic. And so EER induces hepcidin messenger RNA expression and regulates iron metabolism is the take-home. And then they do the next experiments is they do knockdowns with your famous, we all have gone to seminars and we know about knockdown experiments where you use something that will knock down the expression, and that's effectively how they end up proving this. But then they introduce us to the magic of this paper, and they talk about several synthetic ligands have been reported to regulate this receptor transcriptional activity. And they introduce us to the nomenclature GSK 5182, which is a 4-hydroxy-amoxaphen, and they abbreviated 4-OHT analog, is an inverse agonist that specifically inhibits transcriptional activity of this EER. And an inverse agonist, because I always get inverse and agonist because it's sort of like that double negative. This is nothing more than something that binds, that decreases reporter activity through direct binding to the receptor. In this case, it's EER gamma. So this inverse agonist, 4-OHT, ameliorates this Salmonella typhimurium mediated, and now they introduce us to the term hypopheremia, where you don't have enough iron through the suppression of hepcidin expression.
So not enough iron in the serum, but the macrophage concentration of iron is the inverse. It would be higher.
Mm-hmm. Correct. Because Salmonella needs it to grow. So as they take us through this, and this is where we get into another complicated figure 3, is they show us how it ameliorates the Salmonella typhimurium mediated hypopheremia through the suppression of hepcidin. in each one of those panels is elegantly showing us they do everything from hepcidin messenger RNA expression in panel A. Panel B does this really cool in vivo imaging of the hepatic hepcidin promoter, and they use adenovirus expression vectors, one that has luciferase and one that has a hepcidin promoter with a mutation in it. And they inject the mice with vehicle, and you can actually see this expression lighting up in the animals.
Yeah, just to elaborate on that for those listeners who don't have their copy of Nature Medicine in front of them, they lay the mice out on their backs and then can image the whole intact animal and get light where the bacteria are, or where the activity is, rather. So it's a really neat way to see what's happening in a whole living animal.
Right, so they have the hepcidin promoter hooked up to a luciferase, which generates light. So you're basically looking at the regulation of that promoter by the estrogen receptor gamma, estrogen-related receptor gamma, which is a transcription factor, basically. Yeah. And
the hepcidin expression, which is this great homeostatic regulator of, if you will, the mammal that's, locking up the iron, was only observed upon Salmonella typhimurium infection. And so this salmonella-dependent induction of this small peptide hormone, this promoter activity, was significantly blunted. And what they did is they just did a simple IP injection of this inverse agonist, the 4-OHT, whereas the hepcidin promoter, and then they had point mutations. So you can just well imagine the scale of these experiments. They have point mutations in EER, and the EER gamma receptor fail to respond then to the Salmonella typhimurium infection, which then takes us to the fourth figure, which again has 10 little figures in it. And they then teach us through this particular figure, the inverse agonist controls, and this is why they use the magic word at the end, controls intracellular Salmonella typhimurium infection. So that's how they can call it an antibiotic. So the real take-home of this is something that can influence a host receptor can actually have an antibiotic effect on these very difficult infections to treat, because once the bacterium is inside one of our cells, it's very hard to deliver antibiotic into that cell. You need a specific drug that will get transported into the macrophage in order to kill the salmonella. And they, again, take us through. Panel A shows us viable count of intracellular Salmonella typhimurium in the liver, the spleen, again, the medial sinal lymph nodes. And then they show us an immunofluorescence assay of Salmonella typhimurium in neutrophils in our livers, or the mouse livers. And then they take us through the all the other controls that are necessary to drive home the point that this inverse agonist is functioning as an equivalent to an anti-infective, because it's controlling the Salmonella typhimurium infection. So the simple take-home is this EER gamma, which is a major transcriptional regulator that controls hepcidin, is then controlled by the presence or absence, if you will, of Salmonella typhimurium. And this induction of hepcidin expression leading to alteration of host iron metabolism by this bug has enabled them to demonstrate a therapeutic potential of the inverse agonist class of drugs. And, this is, I think, something completely different when you sit through lectures where people are saying we have no new antibiotics, we have no new classes of antibiotics to specifically target structures in the bacteria, But here we're using something in the host. So I found this paper to be elegant, clear. The figures were, really dense when you just count them one at a time. But when you consider the figures from the supplement in concert with the figures, the figurettes from the 4 figures, you effectively have a 60-figure paper. And it's just an incredible amount of work, but it really told the story.
So this estrogen-related receptor gamma, we should probably point out, even though it's called a receptor, it's not a membrane-bound receptor. It's more like steroid receptors, which when they bind their ligand, they go in the nucleus, and then they turn on other genes. So in this case, we don't know what the ligand is for ERR gamma, but in this case, it's induced by the immune response to Salmonella, which produces IL-6, which in turn induces ERR gamma, which in turn induces hepcidin, which controls the iron. So the drug, this GSK drug, basically binds ERR gamma and prevents it from turning on hepcidin. So not enough iron gets in the macrophages, more in the circulation, and Salmonella doesn't grow. It's just beautiful.
Yeah, and we had lab meetings when I was a graduate student with one of the iron labs at Indiana, Gene Weinberg's, and he would always quote this Kipling poem to us when we would go down this rabbit hole of iron, and he made us learn it, and I'll just give you the first few stanzas. Gold is for the mistress, silver for the maid, copper for the craftsman, cunning at his trade. Good, said the baron, sitting in his hall, but iron, cold iron, is master of them all. So he made a rebellion 'gainst the king, his liege, camped before his citadel and summited to siege. Nay, said the cannoneer on the castle wall, but iron, cold iron, shall be master of you all. Woe for the baron and his knights so strong, when the crew cannonballs laid him all along. He was taken prisoner, he was cast in thrall, and iron, cold iron, was master of it all.
Ooh, nice.
I think this Kipling poem really sums up the 2 papers we just did, showing you the importance of this. And you can actually, this should be the exercise for the young graduate students. Where's the macrophage in the Kipling poem, and where's the Salmonella in the Kipling poem? It could be something for the English lit class.
And as a field, we're appreciating how important iron and other nutrients are, and that there's a real battle between the host and the microbe for those essential nutrients. It's called nutritional immunity.
Yeah,
a different strategy by the immune system that we're used to thinking of, but this is also a very different strategy than most antibiotics, which, of course, their specificity typically comes from targeting biological pathways that are unique to bacteria that humans don't have. And now we're talking about a major shift in the iron concentrations in our bloodstream versus the macrophages. It seems a little bit dicey to me.
Clubbish. It's like using a club. Yeah. It's going to be a heavy lift to do a clinical trial. You're going to have to really design a good phase 1 safety trial.
Mm-hmm. Yeah, and then you could imagine side effects of this.
Oh yeah,
compound.
And could you imagine taking it for 2 weeks, or only acutely, or just locally?
Yep. Is this something that might work for Legionella, Michele?
We have actually really good antibiotics against Legionella, so we don't need a new one. Okay. So I would say no, we don't need to go there.
Okay.
And that's because Legionella is extremely fastidious. The iron lab I worked in, my classmate, he was growing, this was in 1980, 81, he was growing up like 4 liters of Legionella, and I was standing next to him, and he was taking the 4 liters to the centrifuge, and one of them dropped.
Yikes.
And broke, and I thought I was going to die. He thought he was going to die.
And who was this?
I'm not going to give his name.
Oh.
You probably know him. You probably know him.
All right. Thank you, Michael. I have a few emails I'd like to share with our listeners. The first is from Ravi, who writes, TWiM and TWiV team, keep up the excellent work. I am an electronics engineer who has never studied biological sciences, but now in my 50s, I find your podcast fascinating. I listen to episodes while working out, a good combination of mental and physical exercise. Thank you. If that's light exercise, then you're helping your plaques. If you haven't already, please read this article in Scientific American, December 2013. It's called Accident of Evolution Allows Fungi to Thrive in Our Bodies. It's an interesting story about Cryptococcus gatti, which is making a surge in the Northwest and other places. And it's infecting people. And they talk about why this is. This is a topic which I have a friend, a former student at the CDC who works on this. Going to have her on sometime to talk about it. But it grows well at 37, which is unusual for a fungus. And it's also, it likes to replicate inside of amoeba. And, that makes it probably help them to replicate in macrophages as well.
Just like Legionella.
Yeah, there you go. So it's a cool story. And Jennifer Fraser is the author, who is a nice science writer that I've read before. To continue with Ravi, what I found interesting, one, fungi have much larger genomes than bacteria or viruses, and this combined with sexual reproduction gives them a larger arsenal for rapid adaptation. Two, in the wild, the biggest predators for this fungus are amoebas. Therefore, it has developed protection mechanisms. If fungus ends up in humans, macrophages look very much like amoebas, and the fungus hitches a ride inside the macrophage, protected from the immune system until it can replicate and do damage. Maybe you can do a show on virulent fungi this week in fungus. Elio should be interested.
Oh, yes.
Yeah, we'll do some shows on Cryptococcus gatti for sure. Anne writes, here's an article which describes a tuberculosis drug that can be tweaked to target multiple disease-causing bacteria and also ward off resistance by targeting multiple pathways in organisms. So this is an article in Inside Illinois. Multi-target TB drug could treat other diseases, and this is in the lab of Eric Oldfield, and the paper is called Multi-Target Drug Discovery for Tuberculosis and Other Diseases. So this is a drug that was originally described with activity against Mycobacterium tuberculosis and also has activity against other bacteria as well. So it's an interesting article to check out.
In the Journal of Medicinal Chemistry.
Yeah. So for those of you who are interested in new drugs, check that out. It's pretty cool chemistry. Jeffrey writes, doctors, I have a suggestion for a slightly out-of-the-way article that would be interesting to hear you analyze. I may be wrong, but this article seems a little primitive and almost designed to continue an alien origin scenario. Despite that, this appears to be a genuine phenomenon in need of some specificity. Personally, I'd love to learn that it is a stratospheric life form that got swept down to Earth, but I suspect that it is much more likely to be an oceanic life form swept up by a typhoon. So this is a weird paper which I have never heard of before. It's called Morphological and Molecular Analysis, calls for a reappraisal of the red rain cells of Kerala. So these are colored particles that fell as red rain over southern India. These are apparently an unidentified prokaryote. Okay, and I don't know anything about this. I emailed Elio to see if he knows anything and if this was worthy of a TWiM at some point. You guys, either of you guys heard of this? The red rain cells of Kerala?
I have not.
Yeah.
No.
Very strange. So check that out. Maybe we'll do it. And I think the same Jeffrey also writes, regarding episode 72, I have to confess that it was one of those episodes that went mostly right over my head. I'm sorry, Jeffrey. Fortunately, I was saved by a listener's question regarding using beer as a vaccine medium against yeast infections. At least I know a little bit about beer.
We all do.
First of all, so I think the question was whether you could immunize people with beer by putting something in the yeast, I suppose, and we didn't know. First of all, I don't have any set answers for your question. I do have some information that might help set the stage. Yeast are indeed used in the production of beer, but comparing the basic beer yeast Saccharomyces with an infectious yeast like, say, Candida is a lot like comparing the stomping power of the Hyrax versus an elephant. They may be related, but they're different beasts. That said, it might be possible and even practical to infect a beer wort with infectious yeasts and have them proliferate. If you could figure out some way to engineer a non-infectious version of that yeast, then I suppose it might be possible to prime the immune system against that. My guess is that the process is a lot trickier than, say, with polio, or people would already be doing it. In addition, it would likely change the taste of the beer, which might or might not be a good thing. Fermentation with non-Saccharomyces yeast is being studied and does have some beneficial properties. Then there's the question of actually getting the non-infectious yeast or some significant immune response inducing part of it into the final product. If you lyse the yeast and the appropriate vaccinating portions were soluble and smaller than the filter size, then that could work if you didn't denature it with the heating process. There might, however, be a simpler way. Europeans and Americans prefer their beer clear and non-yeasty, so they heat it and filter it. There is a class of ancient beers called opaque or sour beers. These are simple beers which are not filtered, hence both yeasty and carbohydrate-y. I don't know if that's a word, at a full meal at lunchtime. They are also produced by the combined actions of yeast and lactic acid bacteria, hence the sourness. For the most part, these beers are living beers made in the home and drunk within a day or two. They're best known in Africa. We
have a brewer of sour beers here in Ann Arbor.
Wow.
Yep, Jolly Pumpkin.
Have you ever had them?
I have.
Are they good?
They're quite distinct.
Yeah. They're cloudy?
They are. And definitely
do they taste
sour.
like sourdough bread?
No. They taste like beer.
Oh, well, that's good.
If the non-infectious infectious yeast could be supplied in a dormant state, able to withstand, for instance, the hot African sun, perhaps in a spray-dried sugar yeast mix, then the yeast could be added to this sour beer, proliferate, and be drunk live to induce the immune response. Any taste differences would be less noticeable in this product, and it would fit in well and easily with the daily food consumption of some areas of the world. Of course, if you could get it in a dormant state in sugar, then it might just be easier to distribute in lollipops. Interesting idea that probably won't work, but it does lead to some interesting possibilities for integrating other medicinal microbes into diets. Well, viruses can be dried into sugar films, and they're much stabler than otherwise. They can last weeks at 60 degrees centigrade when they're dried in these sugar films. So that's certainly something that's being worked on. Anyway, thank you, Jeffrey. I learned a lot about beer. And Dalton writes, heard you mention that you had a competitor by the name of Goggles Optional. So being obsessive about science, I looked them up and tried listening to a few of their podcasts. It's reminiscent of This Week in Science and no real competition to your in-depth science podcasting. Actually, it's a bit aggravating.
Oops.
Sorry about that, guys.
But he doesn't say why.
Yeah, probably it's better. That's
they don't
the
talk
one,
about the weather, maybe.
That's the one that you found, Michele, remember?
Mm-hmm.
Goggles Optional. Someone told you about them, Michele.
Yeah, I think a group out in Berkeley, maybe.
See, people listen to us. And one last one from Juan, who writes, hi, I have some comments regarding your dislike of some choice of words in the microbiology and molecular field. This discussion should fit both TWiV and TWiM. First, you say that the term prokaryote is wrong, but I think the meaning that it has, widely attached to the word, is wrong. Usually people think of the word to mean without nucleus, and usually we think of these organisms coming before the eukaryotes. That is another discussion altogether, but I don't find this is correct. There are two stems, pre and pro. Both of them can be meant to mean before, but in a different way. Pre would mean, to me, something that happens before the appearance of anything. So cells that appeared before eukaryotes would be prekaryotes. Now, prokaryote would mean to me protokaryote, which people may have chosen to shorten to prokaryote. This fits best with the description of these organisms since they have a nucleoid or protonucleus, some sort of organization that is not quite a nucleus, but it is there. So I think prokaryote fits best if it is explained right. Actually, Elio said that he doesn't have any problem with prokaryote. He thinks this whole discussion is not worthwhile. What do you guys think? Michael, do you like prokaryote?
I'm fine with prokaryote. Okay? I think it's what you know it to be. It's like all definitions. You know it when you see it.
By you, Michele?
Yeah. Well, I have heard both sides of the story. So one side who thinks the word is fine recognizes that prokaryote describes the structure, but other people feel it has really clear implications for evolution, and they have strong objections. So I think you need to define it when you use it, maybe in written text.
Mm-hmm.
Are you gonna you use it in your textbook, right, Michele?
Well, we're writing the textbook, so that's something we're going to have to wrestle with.
Yes. The other is your dislike of the expression, a protein is expressed in the X. This is something from TWiV. You say that we shouldn't say this, but instead the protein is translated and only a gene can be expressed. And I think this is wrong as well. A gene is transcribed, the protein is translated. What I mean by wrong is that the word expressed, according to the dictionary, can signify to manifest. Following this, a gene is expressed when the DNA is copied and the specific bases of the gene are put together. An mRNA is expressed when the gene is transcribed by RNA polymerase, and a protein can be expressed when it is translated and when it has reached its final destination. So we're also writing the latest edition of our virology textbook, and we do not use the word express to signify the production of a protein, because we don't think proteins can be expressed. Genes can be expressed, but a protein is synthesized or produced. That's where this is coming from. And many people don't agree with that, and I understand, but it's all, you have to make your definitions and stick by them, especially in a textbook. You can't use a different definition in Chapter 1 and Chapter 10, obviously.
Right.
But the immunologists do.
Sometimes they do, yes. Things change. Things are fluid. Actually, we had an email about that on TWiM. An immunologist, he said, in immunology, you define cells by markers, and then one day the markers change, and so you make more cell types. And that's just the way it is.
And then when you try to track the lineage of the publications back, the nomenclature drives you crazy. So I'm always yelling at graduate students to spell things out rather than use the abbreviations, because in the day of cut and paste, it's not like you have to type the 12 words.
Yep. One other comment is how we are thought about bacteria. It is regarding the fact that bacteria are thought of archaic life forms, and while the first life forms might have resembled more single-cell organisms, both the lineage that gave rise to humans is as old as that of bacteria, since we should have the same primordial ancestor unless there were independent primordial life form formations. Everyone should be taught that any extant organism is as, quote, modern, unquote, as any, since all organisms have to adapt to the changing and current environment. So we can say that current bacteria are the best expression of their lineage, and not that being unicellular organisms, they choose to not continue to innovate biologically. I'm not sure they're the best. They're the ones that are best suited for this time, right? I mean, evolution doesn't produce something that's going to be the best, just what works.
Just a snapshot.
Yeah.
It's what's selected as a consequence of circumstance.
Right. My latest topic of discussion has been the state of science, and it occurred to me that while most people in science say that cutting down the number of PhDs and restructuring, it seems contradictory the fact that there are very active programs of high schoolers and minorities in science with the excuse that we need more scientists. What are your thoughts on this? This is a tough one. Now, it has been suggested that we train fewer PhDs because there's not enough jobs for all of them. But yes, we do tell high schoolers and others we need more scientists because science is what runs the world. But this is a bit of a paradox, and it's largely because the science research budgets in the U.S. have been static for many years. And so it's not fair to have people training with PhDs and not have jobs for them. So that's why that is. But I totally agree that we should be training more scientists, and the budgets should increase by tenfold to accommodate research.
And I'll take that a step further. We don't wring our hands that we have too many high school graduates or we have too many college graduates. So this idea that we have too many PhDs implies that we're some sort of dead end, like all we can do is academic research and replicate ourselves. I think instead we need to talk about the many ways that PhDs contribute to the economy and to our culture and our society.
I would call our listeners to the editorial or perspective that Bruce Albert, Mark Kirshner, Shirley Tigelman, and Harold Varmus wrote that appeared in PNAS in April. It's called Rescuing U.S. Biomedical Research from Its Systemic Flaws. And it really addresses a lot of the issues and the dilemma, but scientists do contribute to society, and the more literate people are about a particular topic, I think the better off society will be.
Yep. And I'll also point out our discussion of that paper on TWiM number 282. And one of the suggestions they make in that paper is one of the main problems that they view, which may be correct, is that PIs often use graduate students to do the bulk of the work in their laboratory, and without them, they would be in trouble. However, if there aren't jobs for them, that isn't quite fair. So the suggestion has been, or one suggestion is, to make the staff scientist a position. This would be a semi-permanent position, someone who stays in your lab for a long period of time and has independence and does a lot of work. And so PIs can rely on that sort of person rather than the PhD students to do all of their work. So, I mean, so you can hear our discussion about that. But I do think, Michele, you're right. We need to train people for other kinds of science careers, not just being PIs, because there are lots of other things to do out there, and I think we could do better on that. And finally, he ends up, thanks for your discussions. I'm always looking forward for each episode of the TWiM VPS and also Urban Agriculture. Thank you, Juan. That will do it for TWiM 81. You can find this episode and all the others at iTunes and also at microbeworld.org/TWiM. And if you want to ask us questions or tell us we did something wrong, we're happy to hear from you. You can send your emails to twim@twiv.tv. Michele Swanson is at the University of Michigan. Thank you, Michele.
My pleasure.
And Michael Schmidt is at the Medical University of South Carolina. Thank you, Michael.
Thank you.
I'm Vincent Racaniello, and you can find me at virology.ws. I'd like to thank the American Society for Microbiology for their support of TWiM, and Chris Gandayan and Ray Ortega for technical help. The music you hear on TWiM is composed and performed by Ronald Jenkins. You can hear his work at ronaldjenkins.com. Thanks for listening, everyone. We'll see you next time on This Week in Microbiology.